Welcome to Life in Biology. I'm Dr. Joel Graff. We're going to do a little lecture here on biotech and omics. Actually, we won't get to the omics part, so just biotech. To start out the class for my Biology 101 course the other day, we started out with a little bit of a review discussing about LUCA, which is the last universal common ancestor. What we did is we reminded ourselves of the three domains of life, at least according to some models, where one branch of life consists of all the bacteria, another branch of life consists of all the archaea, and a third branch of life consists of all the eukaryotes. Those eukaryotes are related to each other, but if you go back in time, you can find a common ancestor at this point right here of archaea and eukaryotes, and then you go back in time further and you get to this line here, which I emphasize by drawing over and over again as the last universal common ancestor. And so what does it mean that all of life today came from a single living species organism, which then split and went different routes through evolution? Well, one of the things that we could mention is that the central dogma of biology that we always talk about, the flow of genetic information from DNA to RNA to protein, that works for both bacteria and humans and everything in between. Another thing that we didn't really get into is the genetic code. This is the ability of the protein making machinery, the translation machinery, to read the RNA 3 nucleotides at a time to make an amino acid sequence, which grew until you get a protein. So the genetic code is the basically like the Morse code of taking genetic sequence of the nucleic acid type and making it into a new type of genetic information, which is of the protein type. So the thing about the genetic code is that DNA from a human could be read and made into protein in yeast or bacteria. My monitor doesn't work well for writing down this corner, but the idea is we could take a human gene and take that genetic information, transfer it into a bacteria, and we could make the human protein product from that gene in the bacteria, because the bacteria knows how to read the genetic code that the humans also use. So all branches of life use the same genetic code. Okay, so biotech is built on DNA technologies. Another term for DNA technologies is genetic engineering. And the way that I like to discuss this is that it's kind of like a word processor where we can cut, copy, and paste whatever sequence we want. We can insert sequences or delete sequences, and we can even do the find and replace bit that you can do with this sequence with a word processor. And so you can change the story over time or mix and match stories, whatever it is you want to do. So we'll run through these one at a time in the following pages, or in the following slides, but you can cut DNA. That means breaking double-stranded DNA. I'm not talking about melting the two separate strands apart. I'm talking about breaking the phosphate sugar backbone that has the covalent bonds. One type of thing that does this is a restriction enzyme, and another system that can do this is the CRISPR system. We'll talk about that in the next slide. Moving on, we can copy DNA. So you can start with a low number of wanted sequence, and then you can amplify it to make as many copies as you have material for. Trillions might be a little bit of a stretch. Billions might be more likely to be what you come up with. So you can start with 1 or 2 sequences, send it through a copy machine. We'll talk about that copy machine, which is called PCR, and you can end up with way more DNA than what you started with, at least of the sequence you want. Then we can paste DNA. So this is when you take nucleic acid, which is a polymer, and you glue two pieces together. So polymer means many parts. So for example, we could have a nucleic acid with 4 nucleotides, 4 parts, and we could have another nucleotide with, we're going to have another nucleic acid with 4 parts and ligate those together, which means glue them together with by removing of water. This is the decondensation reaction we mentioned months ago, and that would give you an 8-part molecule. The insert, delete, and replace, this is used to fix problems or to make new tools. So the first biotech product inserted a human gene into bacteria, and then you have the bacteria make the protein, there should be an E there, and inject humans with that protein. For example, insulin is something that they cloned early on in the biotech days. Bacteria will make the insulin for us, and then you can inject that back into people who are diabetic. Cutting DNA, the two methods we mentioned earlier was the use of restriction endonucleases or restriction enzymes, and the other was the CRISPR-Cas system. So starting with restriction endonucleases, this has each different restriction endonuclease enzyme has a specific sequence that it targets. So for example, I've drawn some sequence here going 5' to 3', 5' to 3'. I drew in the green lines for its phosphate sugar backbone. And then we've got the C's and G's and A's and T's. What I wanted to point out is that as you read this strand in a 5' to 3' direction, I'm highlighting the area here with the red lines. GAATTC is the order of the nucleotides in that stretch of the nucleic acid. And if you go down to the bottom strand, and if we read the bottom strand in a 5', that should be a 3 to 3' direction, it's GAATTC, which should sound familiar because that's what we just said for this here. In other words, this is a palindromic sequence is recognized by restriction endonucleases. That's not always true. Of course, there's always exceptions to the rule, but in this example, we look for a palindromic sequence. The thing about it is that when you have the palindromic sequence, the enzyme is able to cut between between nucleotides in the nucleic acid backbone, and in this case, we have, I didn't mention it in class, but this restriction enzyme that would recognize this particular sequence is called ECHO-R1, and it can cut the DNA between the G and the A. And then if you go down to the bottom strand, you have the G and the A here. It cuts between the G and A there. Again, the A's and T's are only held together with 2 hydrogen bonds each, so it's easy enough to break the DNA apart along that weak link right there. And so I drew up here CCG, GC, or GGC, TTAA. Essentially what I was drawing was CCG and then the bottom line up to the cut point, and then here's the other half of it, and so you break those two pieces apart. Okay, enough of restriction endonucleases. We'll move on to CRISPR. CRISPR is sometimes called CRISPR-Cas because Cas is CRISPR-associated proteins. CRISPR-Cas is the system that is being used these days most often for genetic engineering. And in this case, you only have one enzyme to deal with, whereas you had to get a different enzyme each time you wanted a new cut site over here. And that one enzyme of the CRISPR system can be programmed with guide RNA to cut sequence that matches the guide. Okay, so the green circle here is the protein. It's an endonuclease, meaning it can cut DNA within the middle of a strand. We've got in red here is a guide RNA, and then in black is genomic DNA, and this guide strand and the protein will go around the genome and take a peek all over the place. It'll look at the genome. If things don't match, it'll move on and look at another place in the genome. And if it looks enough times, you might get to a point where the guide RNA will form base pairing with the genome, and at that point, these triangles, these red triangles here, are drawn to show that the genomic DNA would be cut when that happens. Over here, the restriction endonuclease looked for a 6-base pair sequence, and so if you were to just randomly type in G's, A's, C's, and T's on your keyboard, it would, for thousands and thousands and thousands of letters long, you would cut on an average of like every 2,000 or something base pair, whereas because it was just 6 sequences, whatever 4 to the 6th is, over here we've got 4 to the 20th because each nucleotide in the guide RNA would need to match up with the genome, and so the 20 nucleotides that are in the guide RNA give great specificity. The 4 to the 20th is a larger number than you have base pairs in your genome, and so theoretically these should only cut once. In reality, you have something called off-target cuts, and we're not going to talk about that now. Okay, so then we have copying DNA, and there's different ways you can amplify a DNA of stretches that you want, but probably the most common by far is polymerase chain reaction. This is it's a temperature cycling process that doubles your target sequence repeatedly. So with each cycle from hot to cold to warm, and then around those temperatures again, you can end up doubling your DNA. So right here I have some blue DNA, and I highlighted in red where the hydrogen bonds are. And we know that hydrogen bonds aren't that strong, so if you heat a sample up, those hydrogen bonds between the nitrogenous bases that are doing hydrogen bonding here, those are going to break and melt before the covalent bonds in the backbones, phosphate sugar backbones, would break. So you would split that up, and then a polymerase would come in, read the one strand and make a new strand. That's that semi-conservative replication we've talked about before. And let's say you had some green DNA around. Well, you design the experiment so that the reaction will ignore green DNA sequence, and it'll only amplify the blue sequence. Now, doublings going from 1 to 2 doesn't seem like that big of a deal. Then you can go from 2 to 4, and 4 to 8, and 8 to 16. And so you exponentially go up in the number of copies of your wanted sequence. And so you could do doublings of 30 to 40 times. 2 to the 30th, I wrote billions here. Turns out it comes in at just over a billion. And 2 to the 40th comes in at over a trillion. So if you go 30 to 40 cycles, you could theoretically end up with billions, if not trillions. The problem with that is that you don't have enough nucleotides in your initial reaction to make that much DNA. But billion, hundreds of millions, or a billion is plenty most of the time in the lab. We're not that greedy. So then we can go on to pasting DNA. So here, when I talked in the lecture about this, we actually talked about how cells can paste DNA. So I talked about DNA repair machinery. So there's 2 different sets of reactions that can occur in a cell to paste DNA together again after it gets cut. In this first case, I had this black sequence here, and this black sequence here was cut in between. And what happened is we had homology-directed repair, which means that if the cut was, say, on the maternal chromosome, the cell can look over with this strand, look at what the sequence was on one of the strands of the paternal chromosome, and use that as template to fix this strand. So one of these strands would get filled in based on what was going on over here, and then once you had the one out of the two strands, polymerase would come in and fill in the gap on the other side. Okay, so this is recombination is involved in that, where we've got homology going on. So recombination also uses homology. Okay, I'll come back to that later. Now we've got end joining. So we've got a black stretch of DNA and a green stretch of DNA, and you can ligate them together so that the black and green are stuck together. So end joining is completely literal. You just take this end of DNA and that end of DNA and stick them together. Of course, I drew these 5' to 3', 5' to 3', 5' to 3', 5' to 3', so that this 3' end would hook to a 5' end, and this 3' end would hook to this 5' end. And you could glue those together. You wouldn't ever get the 3' ends stuck together or the 5' ends stuck together. You need one 3' and one 5'. Okay. Inserting example. So we're going to use the process of end joining to insert some DNA. We're going to cut, copy, and paste. So we're going to take this sequence, which I've color-coded it black sequence, which ran into blue sequence, which ran into black sequence again. I've cut the sequence right at the boundary on the right side of the blue stretch, between the blue and the black stretch. And then down here, I drew them separate, and then later on, I added in this green DNA here. So if we throw in a piece of green DNA, which, let's say it was a green fluorescent protein gene, we could take that original gene, do the copying by PCR. I got lazy with my writing here. This is double-stranded DNA each. And then you could try pasting that in to the gap that was created when you had the cut here. By doing this, I was giving the example that if there's a promoter region here, a gene that you're interested in here, and you put a green fluorescent protein in here, and then you have the stop codon, you would create a protein there that was, the protein was made from the blue sequence, and part of it would be made from the green sequence. Let's say this blue sequence is a protein you want to study. The green sequence is the fluorescent protein. So wherever in the cell your blue protein wants to go, the green protein would have to follow. And then you can use a microscope and figure out where it is in the cell your protein of interest wants to go hang out. Okay, we're inserting again, cut, copy, paste, but this time we're going to do homology directed repair. So one big difference is that this was in a tube, is what I had pictured in my mind. But let's say you wanted to make a transgenic organism. You could go into that organism with the CRISPR system and cause a cut, and then when you were introducing the components that would make the CRISPR system, you also had some DNA over here that you had done some PCR with, and you could take this sequence that's mouse on the ends and GFP in the middle, and this was supposed to be mouse sequence as well. And because this sequence on the left end, here I drew it double-stranded, so that this line here is drawn double-stranded here. So this green sequence would be recombination, homology-directed repair machinery would be used to patch in the green sequence there, and it would end up getting popped into place. And so that is homology-directed repair, and that's a good way to make some transgenic organism. The patch, you'd want to try to introduce that in, and I'm calling this the patch. You'd want to introduce that at a high number of copies in the cell, so that rather than the cell looking, if this was the maternal chromosome and going over and taking a look at the paternal chromosome to fix the DNA break, you could overwhelm the cell and make it so that chances are the homology-directed repair machinery would take up your patch that you had added into the cells. So that way you don't just go back to having the old sequence you used to have, but instead tip the balance towards the genome taking up the DNA you wanted it to take up. Okay, so a deleting example. So here I've got a stretch of DNA that has some black sequence, red sequence, and green sequence. I could cut the DNA double-stranded break in 2 different places. So there's 4 triangles total, because this is a double-stranded break here and a double-stranded break here. That section in between could float off, and then you could have end joining, glue the black DNA to the green DNA. Okay, so that would be a good way to delete the red sequence. Okay, so then replace using base editors. Something went wonky with my PowerPoint in class, and so here, let's ignore that information. Go back up here. Okay, so we're going to do some replacing now. Most genetic diseases are caused by just a single nucleotide change, and so cutting the whole DNA and inserting a giant chunk or deleting a giant chunk is kind of overkill, and we could go in and do a little bit more nuanced genetic engineering. So here we have again the CRISPR-Cas9 protein, but over here we added on a little extra chunk to the protein known as a base editor. I won't get into it, but it has the ability to change one nucleotide. Here's the genomic DNA in black again, and then in red is the guide RNA. And so let's say that there was a nucleotide here in blue that was causing a genetic disease, and we wanted to change it. Well, by bringing in the CRISPR system, and having this base editor, the base editor would act right on that particular nucleotide, change the sequence, and then the DNA would, the next time the cell replicates, it would change it to the correct nucleotide that you're going for. So you can swap out or modify that nucleotide. And then to finish up, to finish up today's talk, I wanted to just mention that reading DNA is also important. So what's the point of cut, copy, paste, and all that if you couldn't read? So we would want to know the DNA sequence of things to know what we want to cut and paste together. And the first generation sequencing, sometimes called Sanger sequencing, would look at one nucleotide at a time, going past a sensor or on a film. I won't get into it, but a lot of times the upward limit was somewhere around 800 nucleotides. So you'd set up one reaction and you'd get 800 nucleotides of information. Next generation sequencing, or second generation DNA sequencing, really upped the game, and you could get tens of millions of nucleotides of sequence in a single run of the experiment. The problem was that there are short reads, so your tens of millions of DNA bases that you determined were all around 400 nucleotides in length, and so you have 400 nucleotides times whatever to get to 10 million, and that would be the number of different sequences that you'd have to work with. You've got 100,000 different small sequences. Trying to put those together could take a lot of computational power. So that was one drawback to this, is the short reads. So now we've got third generation. This also gives tens of millions, so the number of nucleotides from a single reaction isn't really that much higher, but you get long reads, thousands of nucleotides at a time, tens of thousands of nucleotide per read. So now what the analogy I've heard of is if you had a puzzle that was a certain size of a certain picture and you cut it up into 100 pieces, it wouldn't take most people too long to put that together. But if you turned that same thing into a 5,000 piece puzzle, that would take a lot longer to do all the pairing and matching up of the sequences to see what sequence went next to what. So that is the major benefit of this long read technology, is that you get these long strings. The drawback to the third generation is that you oftentimes get a lot of errors, a lot of nucleotides. It tells you, maybe it says it's an A and it turns out it's a G or something like that. So we can do what's called hybrid sequencing now, which is to combine third generation and second generation sequence. So with the third generation sequence, you can basically build a framework of what you think the DNA is, and then with second generation, each of those pieces would be able to plop down into place because you could match up the color quite fast, and you could make sense of, or you could figure out which of the nucleotides that you got from the long reads were called wrong or were errors, and fix all your errors with the second generation sequencing. Anyway, so that is DNA sequencing in a nutshell. I didn't get to genomics and transcriptomics or proteomics, so perhaps in another lecture. Hope you have a good day.